Thank you so much for inviting me. It's a real pleasure being here. My work is directed at developing data analytics to harness information from physiologic data and to develop dynamical biomarkers capable not only of detecting but also of predicting changes in health status. In this presentation, I will introduce the concept of heart rate fragmentation, a new dynamical biomarker of cardiac autonomic function, and I will describe the applications in MESA, the multi-ethnic study of atherosclerosis, regarding assessment of physiologic changes with aging, prediction of major cardiovascular events and cognitive decline, and the assessment of the restorative effects of sleep on cardiac autonomic function. The development of heart rate fragmentation is possible because heart rate dynamics is primarily controlled by the parasympathetic and the sympathetic branches of the autonomic nervous system. And because with aging and disease, the autonomic control change and imprint dynamical signatures on heart rate fluctuations that we can exploit with the right tools for assessment of health status and risk stratification. In fact, heart rate dynamics RR interval time series provide a non-invasive window into autonomic functionality. I want to call your attention to the fact that the fastest physiologic fluctuations in heart rate are due to vagal tone modulation. What happens is that with inspiration, heart rate goes up, and with expiration, it comes down, defining an oscillatory component that has a name, respiratory sinus arrhythmia. This oscillatory component is the dynamical signature of the coupling between heart rate and respiration. And with aging and disease, vagal tone modulation decreases, and therefore this pattern flattens out. So the HRV, high frequency, short-term variability measures tend to decrease with age and disease. That is, in fact, the paradigm that allows, under which all the HRV analyses are done. So what are the measures of heart rate variability or short-term heart rate variability? The most common are RMSST in the time domain, which basically gives you the average length of each jump in heart rate, and the high frequency power, which quantifies the amount of energy of the signal that is attributable to oscillations that range from 0.15 to 0.4 Hertz, that is oscillations whose cycle length varies between 2.5 seconds and 7 seconds. So really within the respiratory band. And as I was saying, the paradigm for HRV analysis is the fact that higher RMSST or higher high frequency spectral power imply higher vagal tone modulation and therefore a healthier CV profile. The question for us when we start working with MESA was, how does effectively, in actuality, heart rate variability changes with age in MESA? So HRV remains the method of choice for non-invasive autonomic assessments. If you go to PubMed, there's more than 20,000 publications. Let me just, briefly describe MESA. It's a large, ongoing, longitudinal study of the onset of overt cardiovascular disease and the risk factors that predict its progression. It enrolled approximately 7,000 individuals, age 75 plus. And importantly for us at MESA exam 5, Dr. Susan Redline conducted an ancillary study with in-home polysomnography that includes an ECG channel from which we extract all the information for our studies. So as I was saying, how does HRV actually change with age in MESA? And what is supposed is, what we expect is that HRV goes down with age for the reasons that I pointed out. But look what happened in reality. So after age 65 or so, there's an increase in RMSST in these short-term fluctuations in heart rate. We know, and even the standard deviation also increases unexpectedly, standard deviation, the metric overall degree of variability. We know that this is not attributable to vagal tone modulation because it goes down with age and disease. So the question is, what can be inflating these computational metrics, especially in older individuals? So let me show you the ECG of two individuals, one with high, the other with low CV risk scores. Which one is the one with the high, the one with low risk score? I don't think it's possible to say because clinicians themselves label these ECGs as normal sinus rating. But if you look at the time series of the RR intervals, the NM intervals, you will see big differences. So on the left one that corresponds to the top ECG, that is effectively the one from the individual with the lower risk, what you recognize is the respiratory sinus arrhythmia signature, a marker of intact vagal control. The one on the right that corresponds to the middle ECG has a completely different dynamics. It's much more jagged. It's what we call fragmented. And what I highlight is a property that allows us to discriminate these two time series, which is just the percentage of inflection points. That is the instance in which heart rate acceleration changes in sign. So it goes from acceleration to deceleration or vice versa. So is this feature which increases low short-term variability, is this feature, how does this feature changes with aging MACE? And what we see is that it monotonically increases with age. So it appears that this metric will be much more useful than short-term HRV measures because of the non-linearity of the relationship with age. But if that is true, if in fact the percentage of inflection points, the degree of heart rate fragmentation is useful, we should be able to predict, for example, cardiovascular events in MACE. So going from the confounder to the new biomarker, just to summarize, traditional variability measures like standard deviation, RMSST, high frequency power are not reliable probes of neuroautonomic control in older individuals. Why? Because high RMSST or high frequency spectral power values may be attributable to either high vagal tone modulation, which is physiologic and healthy, or to fragmented rhythms. And HRF confounds this traditional HRV analysis, especially in older populations. But as I was saying, our idea was going from the confounder to a marker of severity. So here I show you the Kaplan-Meier survival curves for the prediction of cardiovascular events in MACE over a period of 8 years. And the different lines are for the different quartiles of the measures. Here I have the results for heart rate fragmentation, and in the middle for the Framingham risk score, and on the right for MACE with urinary calcification. And the higher these metrics are, the poorer your prognosis, right? So the more fragmented the individuals are, the higher the lower the probability of survival. So think about how interesting it is that with a single metric, the percentage of inflection points, you can perform in terms of predicting events almost as well as Framingham and MACE, which are composite risk indices that take into consideration age, sex, diabetes, hypertension, etc. It's very interesting. But our point was not to try to propose a substitution for these indices. Our question was whether or not heart rate fragmentation added to these risk indices could augment this predictive value. And that's exactly what we found in MACE. We were happy that in 2021 in Heart Rhythm, a Swiss group confirmed our results in another large population-based study. They found exactly that heart rate fragmentation was predictive and superior to traditional HIV metrics in the association with incidence of cardiovascular events. Additionally, we hypothesized that neuroautonomic impairment quantified by heart rate fragmentation could be associated with cognitive impairment. Why? Because with aging and other CV risk factors are themselves risk factors for cognitive decline syndromes. And also because of the link between vagal impairment and immune dysfunction, the so-called cholinergic anti-inflammatory pathway. Parasympathetic activation suppresses excessive release of cytokines and other mediators of inflammation that may adversely affect central nervous system function. In MACE, we had two time points in which cognitive performance was assessed. At exam 5 and at exam 6, the time window between these two exams is approximately, on average, 6.5 years. And the metric that I'm going to concentrate on, because this is an abbreviated context, right, is the digit span backwards. So this test consists of giving the participant a list, a sequence of numbers, and asking the participant to repeat it backwards. Obviously, the ones that can repeat a longer list have the highest score in this test. And obviously, also, the scores go down with age because working memory, short-term working memory, and executive function decrease with aging. What we also found that is very, very interesting is that the change in score, so the drop in performance from exam 5 to exam 6, increases with your age. So at 8, your drop in score is superior to the drop in score over the same time period if you are 60. So cognitive decline accelerates with age. And we found a similar result with heart rate fragmentation. If you are more fragmented, the probability of dropping of a drop in cognition is superior than if you are less fragmented. So the conclusion is that cognitive decline accelerates with aging as well as with heart rate fragmentation. And I should mention that these results are already for heart rate fragmentation are already considering adjustments for age, sex, and all the other traditional cardiovascular risk factors. So my most recent work was on the association of heart rate fragmentation and brain MRI markers of small vessel disease in MESA. I don't have the time to go through this study, but we found positive and significant associations. Finally, I want to mention that HRF allows us to address a frontline issue, the frontline issue of sleep restoration. We know that sleep is restorative, but the question is, can HRF quantify the restorative effects of sleep on cardiac neuroautonomic control? So our framework is the following. The individuals who benefit the most from sleeping, in whom sleep is responsible for the largest drop in heart rate fragmentation, are the ones with the better cardiac health profile, are the ones with intact neuroautonomic control. And if this hypothesis is correct, we should find that the delta heart rate fragmentation attributable to sleep, so the delta after minus heart rate fragmentation after minus heart rate fragmentation before sleep, this delta should decrease with aging because physiologically aging is not very, everything degrades with aging, and should also be predictive of cardiovascular events. So what we found in MESA actually, as predicted, is that heart rate fragmentation goes up with age, increases with age, both before sleep and after sleep. But interestingly, this delta diminishes with age. This is what I showed here, okay? And so this inability to benefit from sleep, if you want, has also predictive values. If your decline, if sleep is able to lessen your heart rate fragmentation degree, we find that those subjects are protected in terms of future incident events. The more details about this work is in our publication. I just want to thank my collaborators, Dr. Susan Berline, Dr. Susan Eckbert, Tim Hughes, and Eric Berline. Thank you so much.
Thank you so much. We have a question from Brunel University in London. How are individual features other than age, like BMI, relevant when calibrated for when analyzing for the data?
How relevant to the results? No, we control for all those risk factors. We control for age, for differences in age, sex, hypertension status, systolic blood pressure, diabetes, lipid lung medication, smoking. So these results are what you get having adjusted for the traditional CV risk factors.
Great. One more question. Have you studied diurnal patterns of HRF, especially its night patterns?
Patterns are so no, that is ongoing work. So what we did before we analyzed MESA, we also studied the database, the flu database. And what we saw was that heart rate fragmentation tended to be higher during the day than at night. We like the analysis of the night period because activity is controlled for. But in MESA, we now have access to the zero patch data, which monitor heart rate for 14 days. And we will be doing those types of studies.
Wonderful. Thank you so much.
Thank you.